So, summer's over, I guess, officially. Autumn begins either today or tomorrow. I can never remember exactly what it is, but we lucked out, got another beautiful day. It seems like we've had summer the last few weeks here, so it's been great. And welcome everybody to BTI. It's great to see so many people here and such a mix of friends and family and ex-scientists, current scientists. It's really cool. So what I'm going to do today is I'm going to first dim the lights here, if that's okay. Just show my slides. Are we ready to go? Okay. What I'm going to do is kind of give you a little flavor of the history of discovery at BTI over the last 100 years. And what I'm going to do is I'm going to work through each decade and just show you a few photographs and explain some of the scientists and projects that we've worked on over the years. To make it fun, we've also thrown in some trivia questions for you to take a shot at. We're not keeping an official score, so there's no competition, but it's just kind of a fun way, I think. And those trivia questions are really aimed at other things that were going on during those same decades, not necessarily BTI events, but other national or world events that were going on at the time. So let me start first by acknowledging who's worked on this particular project. So Paula, who we just heard from, Aaron Callahan, Mike Carroll, Megan Truesdale, Eric Richards, and the Brandment team that helps us with branding and advertising for BTI have been really helpful in helping us set up this Centennial, since we haven't ever done it. It's the first time we did it. It's the first 100-year history. So we needed help from folks who knew what they were doing for events like this. And so now I'll jump right into the slides. This is a quote from our founder, Boyce Thompson. And here's a picture. Well, you'll see a picture of him in the next slide. And the quote here I'll read, it says, "Any principles concerning the nature of life that you can establish for plants will help you understand man in health and in disease." Is there a strange echo? Is that okay? It's okay? Okay, just checking. I can sort of hear it myself. So the Institute, right, was founded in 1924 by Boyce Thompson. Again, here's a picture of him back then. He was a mining magnate, amassed quite a fortune in mining projects in the United States, especially out in the western part of the U.S. But he also lived in a mansion called Alder Manor, which is on the Hudson River down in Yonkers, New York. And back in around 1917 or 18, I think right around then, he went on a tour of Russia. And at the time, there were famines going on in that part of the world and in other parts of the world. And he realized that food security is a super important thing. And at the time, there really wasn't much research done on agriculture, maybe really none. So he decided to take a big chunk of his fortune and start the Boyce Thompson Institute in 1924. And so this first picture here, we're going to start going through the decades now here. This slide is 24 to 34. This is the founding of the Institute down in Yonkers. See if I can get the little pointer guy to work here for me. There we go. So here's a picture of the Institute under construction. Here it is, the finished version. You can see the greenhouses out here right next to it. There's a golf course. It was a pretty cool spot down in Yonkers where the Institute was, but it's right across the street from Alder Manor, the mansion that I mentioned. And so they say the scientists back then sort of had this pressure early on because he would like to come across the street in the morning to see what was going on in the lab. So he wanted the science close to where he was. This picture here is a dedication ceremony. I think this would probably be on our 100th birthday, so September 24th of 1924. Let me make sure I get these names right here. This is William Crocker, so he's the first managing director, as we call the president today, of the Institute. And then next to him is a man named J.M. Coulter, who was a dean of botany at the University of Chicago, which had connections to the founding of the Institute. I don't know exactly who these guys are. There's a couple of dignitaries from the local area. And then, of course, here's Boyce Thompson. And then standing next to him here is his daughter. And then the last picture on this slide here is this is a picture of the door as you come into the Institute. Just historically, it's interesting. The Institute building is still there, and it was redeveloped maybe a decade or so ago, a little bit longer, into retail space. But interestingly, they saved some of the history of the Institute as well. The building looks kind of similar to the way it does now. The greenhouses are no longer there. But they put a historical room in a lobby of the central part of the building where they have a timeline of the Institute, at least to that point. Eric and I went down to a meeting in the Catskills to help them set up that timeline. And they also put plantings around the Institute, which represent research plants from back then that were being used. So try to save at least some of the history of that particular building. So that's pretty cool. Okay, now, starting our trivia project here, and I need a little help from Paula with the timing. I'm going to give you about 15 seconds for each one. When BTI opened in 1924, there were 24 scientists on staff. What percentage of those scientists were women? Kind of the Final Jeopardy music there. Okay. See, so 38%. It's pretty amazing, really, when you think about it. This is 1924, right? BTI's really had a really interesting history. You know, we finally have a woman president now, Sylvia. That took 100 years. That took 100 years, but we've had a lot of female scientists at BTI over the years, and I'm going to point out a few in some of the other slides here that we're going to look at. Okay, so moving on to the 1930s, here's a picture of a BTI lab at the early institute, and now 1930 to 44, again is William Crocker here on the left, the first managing director. Just to let you all know, you know, BTI has a tradition of having a scientist also being the managing director or the president, and that continues to this day. I mean, Sylvia is an active scientist here and getting her lab set up currently. So we've continued that tradition. And then next to him here is Louis Kunkel, who was a famous plant virologist at BTI early on. And this picture over here on the right is a picture of a commercial product that was developed early on at BTI. It's called Hormodin, is the commercial name for it, and it's still used today. It represents synthetic auxin. So auxin was a plant hormone that was discovered early on at BTI, and it's used to help promote, let's get the pointer thing again here, plant root growth. So it's kind of one of these things where you may have seen it. It has a million different names now if you go to a horticulture store or whatever, or Lowe's or Home Depot. But it's still used today. So this is a very early discovery that's still around with us. Okay, so moving on to our next trivia question. Which American aviator captured the world's attention by becoming the first person to fly solo, nonstop, across the Atlantic Ocean in 1932? Heard a lot of B's. It's D. It's Charles Lindbergh, with the Spirit of St. Louis, I think was the name of his plane. Yeah, Amelia Earhart, I don't know if they keep finding her plane, but anyway, okay. So moving on to the 1940s, one of the things you see on this slide here is this. This is a victory garden that was 1940s. Of course, the war is coming on, or had already started in places for some countries. And it was a time where they had victory gardens, so you're growing vegetables to try to help with the war effort, since everything really had to go into fighting that war. So as a plant research place, BTI, of course, very interested in things like this. In the 40s in BTI, agrochemical development for crop protection was a really important part of what the Institute was working on. It's very interesting because 100 years later, it's a very important part of what the Institute is currently working on. I guess that's not surprising. But the folks that are in these pictures here are, on the left is Wanda Farr, one of the female scientists early on at BTI. And she worked on plant cell development and made a pretty important discovery at the time on the importance of cellulose in plant cell wall structures. The other two guys here in the middle are Hitchcock and Zimmerman, are their names, two other scientists that were working at BTI in the 40s. They developed or discovered a compound called 2,4-D, which was a very important herbicide that's still used to this day. It's a broadleaf herbicide, so it's a compound. It's similar to auxin in that it's a hormone that actually, when you spray it on plants, they start to grow themselves to death. So, it selectively hits broadleaf plants, so that, those are mostly the weeds that you would have in a field of, say, corn or wheat or something which are not broadleaf crop plants. And so even to this day, this compound is still used as a herbicide in a lot of different places. I'll point out that a couple of different labs, especially Frank Schroeder's lab, who's sitting over here, Frank's going to talk to us later today, they're developing, like I said, new compounds that can protect crops in, let's call it a more natural and a little bit less harsh way. And so, like I said, we've got compounds now that the Institute is still working on to sort of better even what we started back in the 40s. And then the last person on this slide, I have to make sure I get it right here, is a guy named Frank Wilcoxon. And I don't want to tell you what he did because I'll give away the trivia question that's coming up. So we'll jump right to that question. And then, in addition to working in plant biochemistry, physiology, and virology, BTI had a team in the 40s working on I heard a lot of A's, and that's correct. Statistics. Frank Wilcoxon developed a statistical model that's used by a lot of different scientists, even outside of plant biology. And so, that's not an unusual thing. One of the great things that have come out of BTI discoveries over the decades are tools that can be used by not only plant biologists, but can be used by other scientists as well. So it's really broad, some of the things that you can do with discoveries that come out of places like BTI. Okay, moving on to the 1950s. Again, another famous female scientist here at BTI. This is Norma Pfeiffer. She worked with lily genetics, so these plants that she's working with here are different varieties of lilies. The picture here in the middle are of special chambers that were used to study air pollution. At the time, BTI has a very strong history of working on environmental air quality kind of things, even to this day. Bob Kohut is still working with the National Forest Services on ways to study ozone levels in different forests. So it's a long part of our history. And then the last picture here is a woman named Clive Chandler, this person right here, who was a famous forest biologist. And here's a picture of her working on some larch trees, actually, which was one of the species that she actually worked on. Okay. Moving on, we're getting close. We're halfway there. In 1952, what scientist made the first X-ray pictures of what would later be termed DNA? Her work, mostly under-rewarded by history at the time, would go on to contribute to one of the most important discoveries in modern biology. Yeah, I hear a lot of C's, and that's right. That's Rosalind Franklin, who worked with the two gentlemen who worked on the DNA structure. She did the X-ray crystallography work to show what the DNA structure looked like, but of course, she wasn't part of that team that got the Nobel Prize for the discovery. That's sort of seen as kind of an oversight at the time. But yeah, she was, and of course, she died of cancer fairly young because she's working with compounds and radiation that they just had no idea back then was as dangerous as it was. So that's kind of a sad side story. Okay, so moving on to the 1960s. BTI research focuses on forest conservation, ecology, and again, working on the effects of air pollution. Two pictures here. A really significant picture, I think, is this one here. This is an early group of high school interns at BTI back in Yonkers. BTI has a 100-year history to this day. Delaney Sickler, who you can talk to today here at BTI, runs our current undergraduate and high school internship program that gives these young kids exposure to science. And I think it's certainly one of the most impactful programs that the Institute's had over its history. And the way it's measured really is you look at where some of these folks have gone. They got excited about science here at BTI. I saw an obituary for somebody who had won a Nobel Prize, and he talked about being an intern at BTI as a high school student. And I've seen it in other places. And if you look at even some of the more recent interns that have gone on to really amazing careers, it's an incredible program and something that Delaney and Megan still work on today. That's her assistant director. The person here on the right is a Austrian-born American virologist. His name is Karl Mara-Morisch. He was at BTI for quite a few years and working on virus transmission in plants. And I'd just kind of like to point out here to remind folks that the first virus that was ever isolated from any organism came from plants, tobacco, mosaic virus. So it's really it's an important plant discovery that's really gone across many different disciplines, and still that way today. Okay, now, let me just preface this question by this is one of the ones where there could be more than one correct answer. In the 1960s, BTI opened secondary research stations in which U.S. states? It's not all of them, but it is these two here, California and Texas. BTI actually owned property in these two states where a lot of forestry research was done, and I'll point out a little bit of that here in just a sec when we look at the 70s. So, of course, 1970s, right? BTI moves to Cornell in 1978, I think was the official year for our move when we became affiliated with the university. New York State helped us build this building here on the left and the one that we're sitting in today, and still helps us with the maintenance of that building. We have a 3-way agreement between Cornell, BTI, and New York State for that move to here. But of course, a lot of research was going on even at the time this move was made. Here's a picture of a guy named make sure I get him right, Jean-Pierre Vité. He was the lead in the BTI forest biology program. And this is a picture of him at one of the forests that BTI owned at the time. I think it's in Texas. I'm not positive, but I'm pretty sure that's where this picture of where he's doing some research is at. Again, I already mentioned in the last slide, this is the under construction of the current BTI building in the 70s. And then this, shoot, hold on. Jumping ahead there. Don't look at that. Okay. Oops. Anyway, this picture over here on the right is a picture of a famous book that came out of BTI that cataloged the estuary of the Hudson River. And of course, BTI's original building overlooked the Hudson River. So that was an important part of ecological research at BTI. Okay. I'll kind of speed up here. By the 70s, the number of volumes in the BTI library was estimated to total. Very good. Everybody got that one. 35,000. So BTI had a fairly extensive library. When you look around BTI today, there's a central area upstairs. We call it the Resource Center now. That was the BTI library. And you'll see there's still a number of books in there, but as everybody knows, things have transitioned to mostly digital, including what we do. Okay, so now we're moving on to the 1980s. And again, I'll speed up here a little bit just for the sake of time. I think I'm going a little bit long as usual. In the 80s, BTI research focuses on how environmental factors impact plants. And there's 3 pictures here. On the left here is a picture of a cabbage loper. So this is a caterpillar that's a common agricultural pest that a couple of different labs at BTI has worked on over the years. And so that's a environmental factor, right? It's a stress. In this case, it's what we call a biotic stress because it's a living thing. Insects are biotic stresses. We also work on what we call, you hear these terms, abiotic stresses. So those are things like air pollution, like we talked about. It can be things like drought, too much water, not enough water, salt, too much salt, not enough salt. So those types of stresses. And so in the middle here is a picture of Bob Kohut and John Lawrence looking at some plants. Sorry, did I get the wrong guy? Bob Amundsen, thanks. He does look like John Lawrence, doesn't he? No, anyway, thanks. Thanks for the correction. And then over here on the right is Carl Leopold, who was a scientist here for many years at BTI. He has an interesting history, of course. He's the son of Aldo Leopold, a famous conservationist, and Carl really kept that tradition going as well locally. Interestingly, he's the founder of the Finger Lakes Land Trust here in Ithaca, and so he was here for many years. His research, one of the interesting things that came out of his research was he worked on these sugars that are in plants that help them to stabilize the plant tissue, and especially when the plants dry out. And so I was involved with him as a tech transfer person on the use of that technology to make inhalable therapeutics. And so, for example, inhalable insulin was one of the products that came out of this work. And if you look now, you can still hear of a number of different, some more successful than the insulin ones that came, and that was really helped by Carl's research. Okay, the 1990s. Things are getting interesting here. BTI embraces new technologies and expands their research scope into the molecular biology of plants and insects. Here on this particular slide, I'm not showing a picture of scientists necessarily, but it's a picture to sort of depict some of the new tools of molecular biology that the Institute was embracing at the time. And so these are dinosaur type pictures now, but this is a picture of a DNA sequencing gel that some of us have been around long enough to have actually done some of these kind of experiments. It's all done much more automated fashion now. On the left here is just a fancy microscope, so microscopic techniques. BTI has a thing now called the PCIC is the acronym. It's so the Plant Cell Imaging Center. Maria Harrison, who works on mycorrhizal fungi, founded that operation here at BTI, and they use special fluorescent microscopes to make these beautiful images of these fungal cells growing, fungal particles growing within the roots of the plants. It's a symbiotic relationship between the plants and the fungus, where the fungus gives nutrients to the plants, and the plants in turn give carbohydrates that the fungus needs to survive. But they use these special microscopic techniques to study those particular things. Okay, moving on to the 2000s, here we are. Establishment of new research expertise in chemical biology and bioinformatics. So everybody understands bioinformatics is really the use of computational, so computers, more in science. And we have a big contingent of bioinformatics folks that are here at BTI. One of the depiction here in the middle, this is a set of servers that are downstairs. This is where a lot of science happens now, are in these computers. It's not quite as large as a Microsoft or a Google server factory you might see out west, but it's a lot of computational space that's needed to do that kind of research, and so that's what that is. Over here is a picture from the cover of Nature when the tomato genome was published. This was a very large consortium of labs, including labs here at BTI and Cornell, that were involved in the genome sequencing of tomatoes. And then over here on the right, I don't want to give away, again, a trivia question, but this is a picture of a virus, and I'll explain what that one is in just a sec. So the trivia question. BTI researcher Robert Granados and his lab developed cell cultures from cabbage loper eggs, so that was the caterpillar insect that I showed you in the earlier slide, in the 90s, and that were eventually named the high five cell line. High five cells were used in the 2000s to produce a vaccine for which disease? Did I ever say D? It's D. So it's cervical cancer. So it was a human papilloma virus that I showed you in that last slide, that blue depiction on the right side. And so at the time that Bob was working on these insect cell lines, he was actually, it's interesting, he was using the cell lines to develop a biocontrol for cabbage lopers. And they were looking for cell lines that would grow these viruses that are natural killers of the insects and hopefully target, a lot of these viruses are very species specific. So they target those, they wanted to target just those pests. And so he was looking for cell lines that grew that virus really well. At the same time, another lab at Texas A&M was developing a way to make recombinant proteins. So that's proteins like a vaccine as an example. We use bacteria to make fairly simpler piles of protein, let's call it. But insect cells were being used at that time. And so Bob went back to his freezer and took out these cell lines that were not so great at making virus like he was trying to do. But it turned out that that second step in this process of making proteins, these cell lines were really good at. And so what happened was the Institute licensed that cell line to a company in Maryland that eventually was sold to this, or sub-licensed to a company called GlaxoSmithKline that developed this HPV vaccine. And so BTI, pretty cool, we're the first insect cell line that was ever approved by the federal government for use in a human patient came from BTI. It actually first was approved in Europe. It's called Cervarix is the name of the vaccine. Folks now are probably more aware of one that's called Gardasil, which is made in yeast but by Merck. But there are two, those were the two HPV vaccines, and it was a huge breakthrough, right? This was, all of a sudden, a discovery was made on what causes cancer, and this vaccine was preventing cancer. You know, so really it's saved millions of lives to today. All right, so we'll run on to the end here. So 2010s, just two more slides, I think. Data-rich omics research, as we call it, expands, including metabolomics. So Frank, again, works on metabolomics. These are the small molecule compounds that he's going to tell us more about today. Proteomics, which are proteins that the genes encode for. And then phenomics, which is kind of a newer term, but BTI as part of the tour. I think you'll get to see the automated phenotyping facility. And just for the non-scientists in the room, a phenotype is really what the plant looks like. And so that's obviously a really important part of the research that we do. We have a couple of other just quick ones up here. BTI recently had 3 National Academy members, Maria Harrison, Jim Giovannoni, and Greg Martin. So we've been really lucky to have some really prominent scientists here at BTI. I mean, everyone is, but they got those special recognitions. The picture here on the right, these are ground cherries, they're called, and Joyce is going to talk about that today, so I won't talk too much about it. Down here on the bottom, Ascribe is a startup company that was founded by BTI recently, and Frank Schroeder and Dan Klessig and members of their lab discovered this technology, and these are small molecule compounds that come from nematodes, which are little soil-dwelling worms. And later, if you want on a tour, I've got some nematodes that you can look at in our translational lab, so you can see where those things come from. But startup companies are one of the business development projects that I personally work on here at BTI. Okay, I'm sorry that went a little long, but